Hello, everyone, and welcome to today's panel presentation titled Identifying Non-Invasive Predictive Biomarkers of Immune-Related Adverse Events Using High-Sensitivity Multiplexed NULISAseq Protein Assays. This webinar is a part of the fourth annual event in the Immuno-Oncology Virtual Event Series. I am Melanie Chan of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by Alamar Biosciences. For more information about Alamar Biosciences, go to alamarbio.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Niyati Jhaveri, Manager of Technology Access Program at Alamar Biosciences, and Jamie S. Lin, Assistant Professor, Department of Section of Nephrology at the University of Texas MD Anderson Cancer Center. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Niyati and Jamie, you may now begin your presentation.
Thanks for the introduction. I'm Niyati Jhaveri, Manager of the Technology Access Program here at Alamar Biosciences, and it's my pleasure today to be talking to you about the highly sensitive NULISA technology. Today, I'll be highlighting a very important study that we did in collaboration with the Cleveland Clinic to identify predictive biomarkers of immune-related adverse events following CAR-T immunotherapy. So we all know proteins are the functional mediators of several cellular processes under homeostatic and pathological conditions. And the blood proteome in particular holds great promise for non-invasive and early identification of many diseases. However, there's several challenges associated with the detection of proteins in blood. And one of the challenges is due to the vast dynamic range of many proteins, from high abundance targets such as human serum albumin, all the way to low abundance cytokines and chemokines that are present in the femtogram per ml range. Current methodologies like mass spectrometry and traditional immunoassays work well for detection of high abundance targets, but they have limited sensitivity for detection of the lower abundance mediators that are some of the key signaling mediators involved in disease pathogenesis and progression. And this is really where Alamar comes in. Our mission is to power precision proteomics by developing highly sensitive proteomic tools that can interrogate the proteome at a deeper level to identify novel markers of disease pathogenesis and to inform the future of precision medicine. The technology that we've developed is the NULISA technology. NULISA stands for Nucleic Acid Linked Immunosandwich Assay and is the only technology on the market that covers all the key dimensions for proteomic analysis. In terms of sensitivity, NULISA provides the best in-class sensitivity in the order of femtogram per ml. The flexible chemistry also enables detection of targets in singleplex format as well as multiplex, up to 250 targets that can be detected together in a single reaction with the same panel. With special tuning strategies, we're able to accommodate the detection of high abundance targets such as CRP, as well as low abundance targets such as IL-6, to cover a wide dynamic range of up to 12 logs. And this is only possible with NULISA and the ability to have the sensitivity, the dynamic range, as well as the multiplexing capability. The fourth pillar of the technology, and the most important one, is really automation. With minimal sample volumes of less than 25 microliters and minimal hands-on time of less than 30 minutes for the assay setup, we can really enable high throughput analysis of many samples within a short amount of time. So how do we achieve that high level of sensitivity? The key really lies in the unique chemistry of the proximity ligation-based assay technology. There's sequential capture and release mechanisms that are built into our workflow that allow us to suppress assay background and enable attomolar sensitivity. So shown here in the schematic here NULISA leverages a pair of antibodies for each target, a capture antibody, as shown in the dark blue here, and a detection antibody shown in the lighter blue. Each of these antibodies is conjugated to a target barcode that's specific to the analyte of interest. And when the antibodies bind to the epitope of interest on the target, there's a proximity ligation reaction that happens, and a reporter is generated that can then be read out by either qPCR for a single-plex assay or next-gen sequencing for a multiplex assay. What's unique about NULISA is that apart from the target-specific barcodes on each antibody, the antibodies also have capture tags. The capture antibody, shown in dark blue here, has a poly A tail, and the detection antibody has a biotinylated oligo tail. And what this enables us to do is to go through sequential rounds of immunocomplex purification to remove all sources of background and achieve really high levels of assay sensitivity. So to go through the workflow step by step, the first step in the reaction is incubating the sample with the antibody cocktail. And this is done in solution where the pair of antibodies bind to different epitopes on the target and form the immunocomplexes. The next step is the first capture step. So we introduce oligo DT beads into the workflow, and via the poly A tail on the capture antibody, the immunocomplex is captured onto the oligo DT beads. The next step then is washing away of any free antibodies, analytes, and matrix, and this allows the retention of the specific analyte bound by the specific capture antibody. In the next step, we can then use a low salt buffer to elute out the captured antibody and analyte complex. The following step then is the introduction of the second capture. This leverages the unique interaction between streptavidin and biotinylated oligos on the detection antibody. And using this unique interaction, we can now perform a second capture step for the immunocomplex of interest. This is followed by a second wash step to remove all sources of background, and what we end up with is a highly enriched immunocomplex that can then achieve the high level of sensitivity that NULISA enables. The final steps in the reaction are the assay readout. And this is done via a proximity ligation reaction that then can be read out by qPCR for a single-plex assay. In the case of the multiplex assay, there's the addition of a sample-specific barcode during the proximity ligation reaction that enables the generation of a single library from all 96 samples in a plate, which can then be sequenced with a standard sequencing workflow. So this is really the unique aspect of NULISA's chemistry, the sequential double capture and release steps that eliminate all sources of background and help us achieve attomolar sensitivity for detections of some of those key mediators that are present in the femtogram per ml range. More information on the sensitivity of the NULISA assay, as well as the multiplexing capability, can be found in our Nature Communications paper from 2023. So I talked about some of the key pillars of the technology, the multiplexing, the sensitivity. The fourth pillar was really the automation. And the automation of the entire workflow is achieved through the Argo HT platform. So Argo has 3 bays that can accommodate 396 well plates and analyze up to 258 samples in under a day. It requires minimal sample input in the range of less than 25 microliters and minimal hands-on time for the assay setup. The flexible chemistry also enables us to transition seamlessly from single-plex formats to multiplex formats. And the Argo instrument has a built-in qPCR machine that can provide ready-to-analyze results in less than 8 hours. For the multiplex workflow, there's a single library that's generated from each plate, which can then be sequenced via an accessory sequencer. So at Alamar, we're really a solutions provider, and we provide the entire gamut of solutions from reagents, instrumentation, as well as software for analysis of the proteomic data. And we're really excited to be partnering up with Dr. Jamie Lin, who you'll be hearing from later in the presentation. Dr. Jamie Lin will be presenting her work using the NULISA inflammation panel to study urine samples and identify biomarker signatures that were associated with nephritis caused due to immune checkpoint inhibitor therapy. So to share some of our internal data, here on the left, we have representative data from a singleplex IL-4 assay showing the best-in-class unprecedented sensitivity that the NULISA platform offers. So in red here, we see results from a traditional PLA, where the limit of detection is in the range of 389 femtomolar. But with the steps of the NULISA workflow that I mentioned before, the sequential capture and release to eliminate all sources of background, we can now achieve limits of detection in the range of 29 attomolar. This represents a 10,000-fold improvement in sensitivity and really enables us to push the levels of proteomic detection beyond what's ever been done before. Apart from the sensitivity, I also talked about our tuning strategy. Our tuning strategy enables us to build multiplex panels that can accommodate detection of higher abundance targets, such as CRP, as well as lower abundance targets, such as IL-4, within the same panel in a single reaction. And so we talked about some of the challenges with the blood proteome, where there's a vast dynamic range of proteins. And now with NULISA and the greater than 12 log dynamic range that our multiplex panels provide, we can really interrogate a larger fraction of the blood proteome. Through the Technology Access Program, we have run several projects ranging from 1 plate to over 40 plates. And consistently, we see high assay precision and reproducibility with less than 10% intraplate CVs and less than 15% interplate CVs. One of the questions that we get a lot is, how do the singleplex assays compare to the multiplex format? And shown here is data for 5 key analytes in both formats. And we see here highly concordant results for IL-4, IL-6, IL-8, IL-1 beta, and TNF alpha, whether it's read with the qPCR readout or the next-gen readout in the multiplex format. What we're seeing here also is that we're able to maintain that high level of sensitivity as well as the wide dynamic range in both formats. And so this really helps us to transition from broader discovery-based biomarker identification studies to targeted validation studies where we can hone down on specific targets and analyze these in a more high throughput fashion. So building on our expertise of developing these multiplex panels, we just recently launched the NULISAseq mouse panel. The mouse panel has been designed for preclinical studies to identify key neurodegenerative and inflammatory markers associated with many different diseases. And through the Technology Access Program, we have applied it to over 25 different preclinical models, including aging, Alzheimer's disease, Parkinson's disease, lupus, as well as colonitis, amongst others. The other multiplex panel that we have is the NULISA CNS disease panel. This panel was carefully curated from feedback from academic leaders as well as pharma and biotech leaders on some of the key pathological markers that are involved in neurodegenerative diseases. What's unique about this panel is that it also allows identification of post-translationally modified proteins like the phospho-taus and phospho-SNCA. Apart from the phospho-tau and phospho-SNCA, there are also other very critical markers such as the myeloid betas, synaptic, vascular, and inflammatory markers that are involved in driving the progression of many of these debilitating diseases. The focus of today's presentation is going to be the NULISAseq Inflammation Panel 250. This panel provides the most comprehensive coverage of cytokines and chemokines on the market and enables detection of 250 biomarkers simultaneously from 25 microliters of biofluid. So today I would like to highlight a study that was done in collaboration with Dr. Neetu Gupta at the Cleveland Clinic. CAR T cell therapies have shown promising results for large B-cell lymphomas, and there are several anti-CD19 therapies, including Axi-cel and Liso-cel, that are FDA approved for the treatment of refractory B-cell lymphomas. However, most of these treatments are limited in their widespread application due to the immune-related adverse effects that accompany CAR T treatment. And the most significant of these IRAEs are cytokine release syndrome and neurotoxicity, or immune cell effector-associated neurotoxicity syndrome. So cytokine release syndrome, as shown here in blue, is really characterized by fever, hypoxia, and an overactivation of the immune system with release of many inflammatory cytokines such as IL-6 and interferon gamma. If left unchecked, this can really wreak havoc and lead to multi-organ failure. CRS is commonly seen in 37 to 93% of patients that are undergoing CAR T therapy and can pose a great health challenge due to the increased hospitalization times and the associated costs. ICANS is seen in 60% of patients and results from memory loss and other symptoms of neurological decline due to the breakdown of the blood-brain barrier and increased vascular permeability. Typically, CRS occurs within a few days of CAR T infusion, and ICANS starts to peak right about the time when the CRS is largely resolved. But in some cases, these two events can occur concurrently, and in extreme cases, it can be very fatal. So understanding these processes on a mechanistic level and identifying the temporal alterations at the proteome level would be very essential to our understanding of CAR T cell therapies and their management. And along with Dr. Neetu Gupta at the Cleveland Clinic, we sought to apply the NULISA inflammation panel to stratify patients that are at the highest risk of developing these IRAEs. So one of the goals was to identify patients that are the highest risk. The other goal was really to identify novel targets that could then be leveraged into druggable targets to develop mitigation strategies and manage the complications associated with these adverse events more effectively. The study consisted of a cohort of 80 patients with large B-cell lymphoma that were treated with anti-CD19 CAR T therapies. Plasma from these samples was collected at baseline and multiple time points after infusion. And based on the clinical grading of the severity of CRS and ICANS in these patients, the patients were grouped into two groups based on the severity of the adverse outcome. So this was essentially a longitudinal study where we collected plasma, 480 plasma samples from patients at baseline, at the day of apheresis, day 0 at the day of the CAR T infusion, 3 time points during the course of the infusion, and day 11 to 16 at the day of discharge. Over 200 proteins were analyzed with the NULISAseq inflammation panel, and linear regression models were used to identify differentially expressed proteins between the low and the high CRS and ICANS groups. Building on the DE analysis, pathway enrichment and network analysis were also performed to identify shared pathways and cytokine hubs that mediate these immune reactions to CAR T therapy. A preprint of the publication is available at medRxiv for further review of the study design. On the next slide here, we have a summary of the overall findings on a more macroscopic level. On the x-axis here, we have the proteins that were identified with NULISA to be significantly different in the groups with high ICANS and CRS scores. The top panel here shows the changes in protein profiles from baseline to day 11 to 16 in the group with high CRS. And we see here that the responses are very early. Starting at day 1, we're already seeing upregulation of many proteins that continues well into day 6 through 9 and 11 through 16. At day 11 to 16, in the very bottom here, there's a trend towards downregulation of many proteins, and some of these are shared between the high CRS and the high ICANS groups. What's interesting to see here is that ICANS also shows early changes with very similar profiles to the high CRS group. But then towards the latter stages, it's really at day 11 to 16 where there's a divergence. We see high levels of downregulation in the ICANS group, and we'll go more into the specific proteins that mediate the clinical manifestations of ICANS. The bottom panel on the slide shows the pathways that are differentially regulated. And it really highlights the power of multiplex profiling. With a comprehensive panel like the NULISA inflammation panel, we can now start to see different biological processes and how these evolve through the course of CAR-T infusion therapy to cause the immune-related adverse effects. The great thing about this data set is that it only requires 25 microliter of plasma and was acquired within a span of 3 to 4 days. So with the NULISA multiplex panels, there's really a wealth of data that you can accumulate to start profiling the changes that are happening at a temporal level. Overall, this is the largest cohort treated with anti-CD19 CAR-T therapy and represents the most comprehensive characterization of the immune response to date. As seen from the heat map before, there were many targets that were shared between ICANS and CRS. And we can see here that there are about 38 proteins that were upregulated in both the high ICANS and the high CRS groups, and 13 proteins that were downregulated in the ICANS and the CRS groups. But there are also several proteins that are unique to each of these phenotypes. And this could reveal further insights into the differences in the underlying biology that contributes to the different time courses and clinical manifestation of these two events. Over the next few slides, I would like to take you through this journey through time as we explore the proteomic changes that are induced after infusion with anti-CD19 CAR-T therapy at day 0. So starting at day 1 to 2, we're already starting to see changes in both the CRS and the ICANS groups. There are about 8 proteins that are upregulated in the CRS group, including IL-17A, CCL2, CXCL8, and IL-34. Some of these are also observed to be upregulated in the ICANS group, such as CXCL1, CXCL8, and CCL2. These are cytokines that are known to be involved in recruiting T cells and other immune cells to the site of inflammation. So what we're seeing here is almost a priming of the immune response to CAR-Ts and pathways such as vitamin D3 metabolism that have been linked to CAR-T efficacy. At day 3 to 5, we see an induction of many proteins with several cytokines reaching peak levels. And the levels are almost 4-fold increase in expression compared to the baseline. This includes known cytokines such as IFNG and IL-6, but it also shows other novel mediators such as CX3CL1, CXCL1, CCL4, and CXCL10, which had not been identified previously in the context of CAR-T toxicity. IL-17 was also overexpressed. It's known to play a role in activation of T cells, but based on the literature and this paper from Rahman from 2018, it's also been implicated in a breakdown of the blood-brain barrier. And so the changes that we're seeing here are almost setting in motion the events that eventually lead up to ICANS and neurotoxicity. Following along the trajectory at day 6 to 9, we see an increase in the Th2 cytokines, such as IL-4, IL-5, and IL-13. These are notoriously challenging cytokines to detect because of their low abundance. And with NULISA, we can now see the temporal regulation of these cytokines in response to CRS and ICANS. What's interesting to note is also that these cytokines are involved in the humoral-mediated response and in tissue repair and CAR-T cell efficacy. So there's really a delicate balance. For CAR-T efficacy, we want to see upregulation of these cytokines. However, uncontrolled upregulation of these cytokines can lead to uncontrolled repair and fibrosis and tip the balance in the direction of toxicity. So it's really important to be able to analyze these proteins in the context of several other alterations at the proteome level, and only a multiplex technology such as NULISA can achieve this level of multiplexing. Along with induction of Th2 cytokines, we also see increased expression of exhaustion markers such as CTLA4, PDCD1, CD274, and LAG3. This suggests that CAR-T cell exhaustion may not only contribute to the decreased persistence and the impaired anti-tumor activity that we are seeing, but these exhaustion markers are also linked with CAR-T toxicity. This finding further underscores the sensitivity of the NULISA platform because these markers have been very challenging to detect in circulation. But with the high sensitivity of the NULISA technology, we can now identify these markers associated with high CRS and high ICANS and start to unravel some of the key biological processes that are mediating these events. And finally, at day 11 to 16, at the time of discharge, we see downregulation of many proteins. So far, we saw an upregulation at day 1 to 2 and massive peaking of cytokines at day 3 to 5 and 6 to 9. But at day 11 to 16, we're kind of seeing a resolution of some of these events. And there's a downregulation of key growth and repair mediators, such as PDGF-A, EGF, and BDNF. BDNF is brain-derived neurotrophic factor. It's the most prevalent growth factor in the brain, and it's involved in neuronal development and plasticity. PDGF-A is important for organ fibrosis and vascular integrity. And so what we're seeing here with downregulation of these key cytokines is the events that lead up to the breakdown of the blood-brain barrier and loss of vascular integrity that cause some of the clinical manifestations of ICANS in terms of cerebral edema and vascular permeabilization. So to summarize, with the highly sensitive NULISA technology, we were able to comprehensively characterize the temporal response to CAR-T therapy in patients with cytokine release syndrome and ICANS. The changes in the plasma proteome occur very early as day 1 to 2 after infusion, followed by peak induction of Th2 cytokines and exhaustion markers. 2 weeks into the infusion, there's a massive downregulation of proteins involved in tissue repair and vascular integrity, suggesting a really well-orchestrated sequence of events that lead up to the disruption of the blood-brain barrier and cerebral edema that's seen in severe cases of ICANS. On a pathway level, we also saw many shared pathways between the high CRS and the high ICANS group. We see upregulation of inflammatory pathways, IL-17A, as well as vitamin D3 pathways that have been linked to CAR-T efficacy previously, but are now being implicated in CAR-T toxicity. Interestingly, we also saw some differences, particularly in the downregulation of TNF pathways that was exclusively seen in the ICANS group. This is in stark contrast to the upregulation of the TNF pathway that's seen in CRS. And so these differences in the pathways between the CRS and the ICANS group suggest differences in underlying biological mechanisms that regulate these events and their temporal course. Apart from known markers such as IL-6 and IFN gamma, the NULISA multiplex panel was also able to identify novel mediators that had previously not been described in the context of CAR-T toxicity. And we're really excited to be exploring these further along with the Cleveland Clinic team. So apart from the known markers, the breadth of the NULISA panel really enabled identification of novel immune mediators. With the high sensitivity, the multiplexing capability, and the high throughput offered by NULISA on the Argo platform, we're excited to support the entire gamut from discovery-based studies to translational target validation studies to enable identification of predictive signatures, such as the ones discovered here in the context of CAR-T toxicity, to empower future diagnostic applications and inform precision medicine. With that, I'd like to thank Dr. Niyati at Cleveland Clinic for their close collaboration, and I look forward to addressing questions towards the end of the session. Next, we have Dr. Jamie Lin from MD Anderson talking about urine proteomics and how, leveraging the NULISA panel, they were able to identify signatures associated with nephritis.
Hi, I'm Dr. Jamie Lin. I'm an onconephrologist at MD Anderson. I'm excited to be here on Labroots today to share some of our group's recent work. Today, I'll be talking about how urine proteomics reveals a novel, non-invasive diagnostic signature for the diagnosing immune checkpoint inhibitor-associated nephritis. I'll begin with the background on immune checkpoint inhibitors and their associated side effects, or immune-related adverse events, the clinical challenge that we face with diagnosing acute interstitial nephritis, or AIN, and how we're leveraging urine proteomics for biomarker discovery to help us with this clinical challenge. I'll end by telling you about the implications for clinical care using our key findings. So immune checkpoint inhibitors are a transformative class of cancer therapies. By blocking inhibitory pathways such as CTLA4, PD1, PDL1, they release the natural brakes on T cells, enhancing anti-tumor activity. But these same mechanisms can also trigger off-target immune responses, leading to autoimmune complications. Since the mechanisms that underlie anti-tumor immunity and autoimmune side effects are the same, we actually see a high percentage of patients develop these immune-related adverse events, which are essentially autoimmune complications. So roughly 60% of patients on immune checkpoint inhibitors will develop an IRE. And because this is a sign that a patient's actually responding to immune checkpoint inhibitor therapy, early detection of these side effects enables us to intervene and then continue with the therapy. In the more common IRAEs like colitis, dermatitis, and endocrinopathies, they usually present with clinical symptoms or they have a blood-based diagnostic test. Less common IREs like immune nephritis or AIN, unfortunately, doesn't present with clinical symptoms and doesn't have a blood or urine-based test to tell you that this is AIN. And unfortunately, AIN is associated with high patient morbidity. So what exactly is the challenge of AIN if it's not that frequent of a side effect? While only 2 to 5% of patients will have immune checkpoint inhibitor-associated AIN, approximately 20% of patients, or 1 in 5 patients on ICI therapy, will develop kidney injury. And so actually the goal is to figure out of these 20% of patients, or 1 in 5 patients who have AKI, which of those actually have true AIN or immune nephritis. And because we don't have a diagnostic blood or urine test, we rely on an invasive kidney biopsy. And so this is kind of what the current diagnostic pathway looks like. It's fairly time consuming and it's invasive. So when we see a patient on immune checkpoint inhibitor therapy and they develop kidney injury, our first goal is then to evaluate for potential causes for their kidney injury. Is it because they have low blood pressure, there's an infection, they're dehydrated? And if we're able to reverse this and their kidney function or their serum creatinine improves, then we can start back on immune checkpoint inhibitor therapy. But if the kidney function doesn't improve, then now we're looking at getting a kidney biopsy, and that takes time. And it's not feasible for all patients with cancer. And then once the biopsy is back, then we can treat based off those results. But essentially what I'm showing you here is that AIN or immune nephritis is a diagnosis of exclusion. So there are several reasons why a biopsy may not always be feasible. You need a skilled proceduralist in order to do this procedure. The patient has to be able to lie prone. So if they have advanced disease, this might not be possible. Scheduling takes time. Patients with cancer probably have already undergone a lot of invasive procedures and don't want to add another procedure onto their to-do list. But most importantly, the risk of bleeding is actually higher in patients with cancer because they might have anemia, thrombocytopenia, coagulopathy, or they might be on another medication such as the tyrosine kinase inhibitor that increases their bleeding risk. Unfortunately, that means that a lot of times those patients with true ICI-AN are going to have delayed diagnosis. So by the time they are actually diagnosed and then treated with steroids, only 40% of these patients will recover full kidney recovery. And that means an overwhelming 60% of patients will have compromised kidney function after this disease. And while it might not seem like a big ordeal, it actually is for patients with cancer. because it basically means that they may not be able to go back on immune checkpoint inhibitor therapy, but it might also limit the opportunities that they have for other cancer therapies down the road. As the eligibility of ICI therapy expands, so does the burden of immune-related adverse events. And so there's a critical need for a non-invasive diagnostic test that supports timely, personalized treatment decisions for immune nephritis. Our group has demonstrated that urine provides a unique, non-invasive window into the kidney's immune environment in immune nephritis. And other research groups are pursuing similar approaches, which speaks to the urgency and the importance of this question. However, to date, there's still no consensus on an optimal biomarker. One of the major challenges in identifying a urine biomarker has been the limitation of existing platforms. So conventional immunoassays like bead-based immunoassays or proximity ligation assays lack the sensitivity and multiplexing capability to be able to capture the wide complex immune signals driving acute interstitial nephritis. To address these limitations, our group leveraged NULISA, a highly sensitive assay that detects low abundance proteins and supports multiplexing of over 200 targets in a single run. This technology allowed us to comprehensively profile immune signals in the urine and in the plasma. In our recent publication in JITC, we investigated the urine and plasma profiles of patients who developed acute kidney injury on ICI therapy. We enrolled patients between July 2020 and January 2023. We then grouped them into those with biopsy-proven ICI-AAN or non-AAN. And in this specific research focus, we were very careful about the patients that we were going to exclude. So we excluded cases where the patient was already more likely than not to undergo a kidney biopsy. So if their urine sediment was active with blood or proteins, such as in the cases of glomerular nephritis or vasculitis, we excluded those cases. And of course, if a patient was already started on treatment for ICI-AAN with steroids, which is going to impact the protein levels in urine and plasma, we also excluded those cases. And so that left us with a total of 49 cases, 22 patients with ICI-AAN and 27 with non-AAN. So we evaluated the clinical differences in our ICI-AAN group versus our non-AAN group. In our immunonephritis group, what we saw was that these patients were more likely to have a positive urine eosinophil test, a higher urine protein to creatinine ratio, even though that was still less than 1 gram, as well as a higher AKI or acute kidney injury stage. They also were more likely to have a higher CRP level. These features all reflect a heightened immune activity, but they're not specific enough for diagnosis. So we started by analyzing all the urine and plasma samples from both groups, the ICI-AAN group and the non-AAN group. And we used clustering and dimensionality reduction analysis. And what we found was that there was a clear separation in plasma and urine profiles. So plasma in the red and urine in the blue. And you can see that they don't overlap, which means that they're not redundant sources of information. Instead, what we suspect is that they're actually offering complementary information. So the immune activity at a systemic level versus at a tissue level. Of the 203 targetable proteins, we were able to detect over 73% of those proteins in urine and over 95% in plasma. And then when we investigated for those proteins that were differentially expressed in only immune nephritis, we found that there were 95 differentially expressed proteins. 59 were identified to be unique in urine, and that's represented by the blue dots, and 22 unique to plasma, so those in the red dots. There were a total of 14 proteins that overlapped between urine and plasma. And when we looked at those 14 proteins a little bit more closely, what we saw was that there was a bigger fold change in urine compared to plasma, suggesting that urine not only outperforms plasma, but it offers greater disease signal. We investigated the molecular mechanisms that might underpin immunonephritis, and the top pathway came back as cytokine receptor interaction. But what we also identified was that JAK-STAT signaling pathway as well as TNF signaling pathway were involved, suggesting that you can therapeutically target these pathways for treatment of AIN. We already used infliximab, which is a TNF inhibitor for patients with AIN, and we have a single case report where we use a JAK inhibitor treating a patient with refractory AIN. So there's more work to be done here. But all in all, this initial data led us to focus more on the urine proteome as a specific source of biomarkers. So we had over 60 proteins that were differentially expressed in urine, and we wanted to narrow this list down. So we applied some stringent filtering criteria for the proteins of interest. We looked at only those proteins that were statistically significant with an adjusted p-value of less than 0.01, an area under the curve or an AUC of greater than 0.8 for being able to differentiate ICI-AIN from non-AIN, as well as those proteins that had a large difference in their fold change, so greater than 8. We ended up identifying 10 key analytes. And of these 10 key analytes, CXCL9 has been previously identified by our group and others, as well as CD274 in tissue. And that means that the rest of these 8 proteins are novel proteins that haven't been previously reported to be associated with immunonephritis. And overall, these proteins have functions in chemotaxis, hypersensitivity, immune activation, immune regulation, and apoptosis, all suggesting a heightened immune activity that's going on at the kidney level. So even with 10 key markers, that's still a lot for us to work with. So we enlisted the help of Dr. James P. Long, our biostatistician that's worked with us for several years now. And together we applied statistical and machine learning techniques to identify a specific marker or markers for AIN signature. Using penalized logistic regression, which is a method for selecting small sets of features with high combined performances, we identified 5 markers. And then using forward stepwise selection and all subset selection, we identified an additional 3 markers using forward stepwise selection and 2 using all subset selection. And what you'll see from this analysis is that IL-5 and FAS continuously show up as top markers for being able to differentiate cases of ICI-AIN versus non-AIN. To assess the robustness and interpretability of IL-5 and FAS, we performed bootstrap resampling to evaluate the model stability, and IL-5 and FAS consistently emerged as the most frequently selected features, appearing in over 80% of the bootstrap iterations. And then using CART, or classification regression trees, to identify potential nonlinear relationships, again, FAS and IL-5 were selected, suggesting that IL-5 and FAS are the top discriminators of ICI-AIN versus non-AIN, and reinforcing their diagnostic value as a biomarker signature. Our analysis culminated in evaluating the diagnostic performance of these key biomarkers using ROC curves. And what we found with looking at each of the individual markers, IL-5, FAS, and CXCL9, was that their AUC was roughly at 0.8, whereas the signature of IL-5 and FAS gave us a better AUC of 0.94, or 94% accuracy. When we took a look to see why the signature did so well, we saw that IL-5 and FAS were actually complementary. IL-5 gave us a sensitivity of 100% with a specificity of over 50%, whereas FAST gave us a specificity of 100% and a sensitivity of over 50%. And then when we assess the robustness of this signature using 5-fold cross-validation, the AUC remains stable, ranging from 0.9 to 1.0. And on the right, the scatter plot shows us the IL-5 and FAST level across all the cases. And the black line indicates the decision boundary where the model assigns equal probability of either ICI-AAN versus non-AAN. And you see that the majority of the blue dots where the ICI-AAN cases are above the black line, whereas the majority of the non-AAN cases are below the equal probability line. What we're working on right now is validating this signature with an external cohort. And where we see this going is that there's clinical application for using this IL-5 and FAST signature. So in the future, what we envision is that when a patient on immune checkpoint inhibitor therapy develops acute kidney injury, we can check a rapid urine IL-5 and FAST test and hopefully have those results within a day or two. This allows for faster diagnosis. It avoids the need for unnecessary kidney biopsy, and actually the majority of patients who develop AKI. It minimizes treatment interruptions, and potentially it's also going to improve kidney outcomes for those patients that really have AIN, because we'll be able to diagnose and treat them faster. In summary, using NULISA, we developed a novel and non-invasive urine signature for ICI-AN diagnosis that consists of IL-5 and FAST that achieves a 94% diagnostic accuracy. And our analysis also uncovered several novel urine protein markers and potential targetable pathways that haven't been previously reported. The next steps for us are external validating our signature, exploring potential targeted therapies such as JAK-STAT inhibitors, and development of point-of-care testing. Clinically, the work that we've done here and identification of this signature means that we can streamline diagnosis of patients with ICI-AAN. We can reduce biopsy reliance and help maintain the continuation of life-saving immune checkpoint inhibitor therapy for patients with cancer. And with that, I'd like to acknowledge all of our patients, collaborators, research team, and support who made this work possible. I'd like to thank everyone and remind everyone that with urine, you're in good hands.
Thank you, Niyati and Jamie, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. Let's get started. So it looks like we have quite a few questions that already came in. The first question asks, what is the future direction of this work?
So I can take that question. So our next step is to validate this signature of IL-5 and FAST. We'd also like to study how the signature changes when a patient's on treatment. Do the levels go down? Does that correlate with improvement in their kidney function? And if we wanted to re-challenge these patients on immune checkpoint inhibitor therapy, can we also use this signature to monitor how well they're doing from a kidney perspective? Of course, it's not just with ICI therapy. AIN can be caused from other etiologies, and so looking at AIN from different triggers would also be something that can be done.
Awesome, thank you. Another question did come in. I don't know if I'm going to be asking this correctly, but it says, not being familiar with the progression and details of ICI-AIN, what kind of testing regimen or diagnostic testing can you imagine? One-time, surveillance, etc.?
So I'm not sure if I am fully following this question, but I guess if So right now, when we see a patient who develops acute kidney injury on immune checkpoint inhibitor therapy, we check our basic standard urine and blood-based markers, and we try to reverse whatever stress might be attributing or contributing to the kidney dysfunction. If we were to use this signature, I can envision a future where we say, hey, if a patient has a high IL-5 and high FASN level, we should go ahead and biopsy and treat the patient. And while the patient is on treatment, we can collect sequential, biomarkers of IL-5 or biomarker levels of IL-5 and FASN to say, oh yes, they are responding, go ahead and continue treatment. And if the levels drop quickly, maybe they don't need to be on treatment for the full 3 to 4 weeks and maybe just 1 to 2 weeks, whereas someone with levels that aren't coming down might need longer treatment duration with steroids.
Okay, great. The next question asks, how much urine is needed for the NULISA assay?
Jamie, did you want me to take that one?
Yes, go ahead.
Yeah, thanks for the question, Alex. So the NULISA assays require minimal sample volume. Each assay requires about 25 microliter of sample, and this accounts for the dead volume requirements on the Argo platform as well.
Okay, great. The next question asks, what other sample types have you tested on the NULISA system?
Mm-hmm. Yeah, that's a great question. So given the high sensitivity and the wide dynamic range of NULISA, it's really broadly applicable across many different sample types. And you've heard from Jamie about testing in urine samples. We've also tested the standard matrices, plasma, serum, CSF, but also non-invasive sample types such as saliva, nasal swabs, and sputum. It's also ideally suited for low volume samples. And we had a poster presentation recently at EDPD highlighting high detectability of neuromarkers like phosphotau's and GFAP and NFL in dried blood and dried plasma spots. So there's a lot of interest in that particular application for self-sampling. And we're looking forward to working with the research community.
Great. The next question that came in asks, have you looked for signature of IO response using the Alamar system?
So I can take that question. So one of the current struggles of IO therapy is actually identifying if a patient is responding to treatment, because oftentimes it can take actual months before you start to see changes by imaging. Our group previously looked at TLS or tertiary lymphoid structure signatures, which is a marker of response to immune checkpoint inhibitor therapy. We found this signature in both the tissue as well as the urine of patients who developed nephritis. So if you were to extrapolate this backwards and use, a TLS signature or maybe just a marker within TLS, let's say CXCL13, theoretically, you could use Alamar and NULISA to check these levels from the beginning of treatment, because you're able to detect pentagram per milliliter over time, and see that the levels are going up. And this could be done way earlier than you can see changes on imaging.
Thanks, Jamie. The next question that we received asks, are there other immune-related adverse events that you could address with inflammation panel?
I can take this question as well. I think there are, nephritis is unique in the sense that we have urine to have a direct window into the kidney and what's going on. For patients who develop pneumonitis, also very high morbidity, high mortality, IRE, you could use pleural fluid to look at signatures in the pleural fluid. But for other IREs, like let's say myocarditis, you're not going to have fluid per se. However, you're still going to see systemic inflammation markers that are circulating in the serum. So while it might not be 100% organ specific, it's possible to create a panel that is able to say, hey, for sure, this is an immune-related adverse event, and we're suspecting this is myocarditis.
Great. So it looks like we're down to our last two questions. The first one asks, how do you compare to other platforms in terms of sensitivity?
Mm-hmm. Thanks, Melanie. So NULISA really has the best-in-class sensitivity, and this is largely due to the unique chemistry that we were describing before, with the sequential capture and release steps that really purify the immunocomplex and eliminate all the major sources of background. So in our Nature Communications paper that I highlighted earlier, we did a cross-platform comparison looking at different other platforms, proximity extension-based assays, as well as single molecule array technologies. And NULISA consistently outperformed all the technologies in terms of sensitivity, with almost a 10,000-fold improvement in overall sensitivity.
Great. And the last question we have asks, how much sample do you need for the mouse assay? Have you validated low input amounts?
Mm-hmm. Yeah, so with the mouse panel, we've tested it in over 25 different preclinical models so far. These have mainly been with plasma, CSF, interstitial fluid, as well as brain lysates. And we recognize that CSF sampling in mice can be pretty challenging. So we've tested multiple dilutions of these samples, and even with twofold dilution of both CSF and plasma, we're still able to maintain high detectability in the range of greater than 95% for plasma and greater than 75% detectability for CSF.
Great. Thank you, Niyati and Jamie. Do you have any final comments for our audience today?
I'm personally really excited to see where NULISA's platforms are going to take us. This is really quite a breakthrough to be able to capture such a wide range of proteins. And, in the world of nephrology, we've been trying to do urine analysis for a really, really long time and have not been as successful as we would like to be. So I think we're going to see a lot of changes and improvements in the nephrology world with this assay.
Yeah, it's been great to work with collaborators like Dr. Lin and then Dr. Henrik Zetterberg in the neurospace. We're excited to, see what insights NULISA can provide.
Awesome. Thank you again, Niyati, Jhaveri, and Jamie Lin for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Alamar Biosciences, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2026. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.